Hi, this is Dr. Neela Bakore. We are done with the first chapter of the unit of cell. The chapter is Cell, the unit of life, where we discuss the complete structure of cell, the cell organelle and everything. And now we are planning to discuss all the questions which are given in our NCERT book. So let us take these questions one by one, and the basic idea is to understand which all important points should be there in our answer so that we are able to score 100% marks. So let us take the first question in this particular chapter. The chapter that we are taking is Cell, unit of life. The first question reads, which of the following is not correct? So our main emphasis should be something which is given to us is not correct or incorrect. They have given us 4 points and they want to know that which statement is incorrect. The first one is Robert Brown discovered the cell. This is an incorrect statement because we know Robert Hooke saw the cell for the first time or discovered the non-living cells, and Leeuwenhoek discovered the living cell. So A option, that is Robert Brown discovered the cell, this is incorrect because it was Robert Hooke who discovered the cell. So I think we got our first answer, which is not correct, but let us see the other options also. B says Schleiden and Schwann formulated the cell theory. This is correct. So B statement is a correct statement. Virchow, C statement says Virchow explained that cells are formed from pre-existing cells. If you remember, we talked about the cell theory and Virchow added one point to it. He said omnis cellula a cellula. That means he was the one who actually gave the concept of that cells arise from pre-existing cells. So statement C is also correct. D statement is a unicellular organism carries out its life activities within a single cell. One cell like amoeba or any prokaryotic cell, they are capable of performing all activities like respiration, digestion, excretion, reproduction, everything. So statement 4, that is D, is also correct. So our question was which out of the 4 statements is incorrect. So the first one, this is incorrect. So A is incorrect. Coming to question number 2. New cells generate from. Question number 2. New cells generate from. 4 options given again. Bacterial fermentation, which is not possible because fermentation helps in production of ethyl alcohol. B, regeneration of old cells. Normally old cells are not capable of reproduction, they have lost their power of reproduction. C is pre-existing cells. Rudolf Virchow said omnis cellula a cellula, that means they arise from pre-existing cells. So here C option, which says the cells arise from pre-existing cells, is the correct one. So our answer is going to be new cells generate from pre-existing cells, that is C. The third question is about match the following. They have given us 2 columns. So column 1 and column 2. Column 1 has 3 things, A, B and C. One is Christie, then is Cisterni, and third is thylakoid. And in column 2 also 3 things are given. The first in column 2 reads flat membranous sacs in stroma. We know thylakoids are those simple stack like things which get stacked to form grana. So 1, that is flat membranous sac in flat membranous sac in stroma. This would be matching with thylakoid. So what is thylakoid? Thylakoids are flat membranous stacks which are found in stroma of chloroplast. Second point in column B says enfoldings in mitochondria. Mitochondria structure we have seen in details. The outer membrane is smooth, the inner membrane has, so if I draw a little mitochondria here, maybe the outer membrane is smooth and this inner membrane is thrown into these folds. These folds are known as cristae. So the second one here is for crista, that is enfoldings in mitochondria. Enfoldings in mitochondria. So this is for crista and this is for thylakoid. Now the third is in column 2, disc shaped sacs in Golgi apparatus. So when we make these Golgi apparatus, we make these kind of flat tube like structures which are known as cisternae. So what are cisternae? These are, so here the third one is for cisternae, that is disc shaped. Disc shaped sac or sacs in Golgi. So the options would be A with 2 in column 2, that is enfolds, cisternae with 3 in column 2, that is disc shaped, and C thylakoid is with 1 flat membrane which are sac like structures in the stroma. Now let us take question number 4. Question number 4 is again 4 statements given and we have to identify the one which is correct. Which of the following is correct? 4 statements are given. First statement is cells of all living organisms have nucleus. Is it correct? It is not, because prokaryotic cells do not have nucleus, they have nucleoid. Secondly, there are some eukaryotic cells like mature mammalian RBCs, sieve tubes or sieve cells, they are also enucleate when they mature. So A statement is incorrect. We have to find the correct statement. Out of 4, we have to pick the correct one. So A statement which says cells of all living organisms have a nucleus is incorrect. So this is incorrect. Let us talk about the second one, B. B says both animal and plant cells have a well defined cell wall. This is also incorrect because this is the technical difference between a plant cell and an animal cell. Plant cells have a cell wall outside plasma membrane whereas animal cells don't. So this is also an incorrect statement. Third one, C statement says in prokaryotes there is no membrane bound structures. This is correct. In prokaryotic cell we have only outer plasma membrane. Inside there is a nucleoid, free ribosomes. There are no membrane bound structures, which means organelles are absent. So this is a correct statement. Let us also read the D statement. It says cells are formed de novo from abiotic material. Here we need to understand the meaning of word de novo. De novo means every time formed new from something as raw material. So are all cells formed every time? We know in the origin of life the cells developed from abiotic things, but that was the first cell which was formed. After that all cells are coming by division of the pre-existing cells. So statement D is also incorrect. So our question was to pick the correct statement out of the 4, that is C, which says in prokaryotes there are no membrane bound organelle. Fifth question. Question number 5. What is mesosome in a prokaryotic cell? Mention the function that it performs. What is mesosome? When we discussed prokaryotic cell, we made the bacterial structure or a bacterial cell. We also made cell wall outside it of peptidoglycan, and at some places we showed that this plasma membrane shows infoldings. These infoldings of plasma membrane are known as mesosomes. So what are mesosomes? Mesosomes are infoldings of plasma membrane. That is the structure. Now the second part of the question says, mention the functions that it performs. Mesosomes have respiratory enzymes. They have as they have respiratory enzymes, they help in respiration or respiratory process. The structure in eukaryote which performs the same function is mitochondria. So functionally they are similar to mitochondria of eukaryotes. So our answer should be that mesosomes are infoldings of plasma membrane which have respiratory enzyme. Their function is to help or perform respiratory processes, and they function similar to mitochondria of eukaryotes. Question number 6. Sixth question is, how do neutral solutes move across the plasma membrane? See, whenever we answer a question, we have to keep in mind how many parts are there, because for evaluation purpose or for marking things, every part of a question has been assigned some marks. So if we miss one part, we are not going to get marks for that particular part of the question. So here the first part is, how do neutral solutes move across the plasma membrane? Can the polar molecules also move across it in the same way? This is part 2 of the question. And third one says, if not, then how are these transported across the membrane? So three parts of this question which we need to answer. First is, how do neutral solutes move across the plasma membrane? So this is part 1 that we are talking of. Neutral substances, they can simply cross the plasma membrane by diffusion. Neutral substances, they cross or they move by diffusion. We know diffusion is a passive process. It moves along the concentration gradient. So these substances will be taken in when the concentration of these particles is more outside the cell and less inside. So they will move from higher concentration to lower concentration. It is a passive process. So our first part should have points like neutral substances cross the plasma membrane by simple diffusion, which is a passive process, and these particles are moving from their higher concentration to lower concentration. Passive means there is no expenditure of energy. Now the second part of this question is, can polar molecules also move across? What is meant by polar molecules actually? The hydrophobic, hydrophilic, these kind of molecules. Can the polar molecules also move across the membrane in the same way? The answer is no. Reason? This is for the first thing. The second part, polar molecules cannot move by diffusion. So polar molecules do not cross the plasma membrane by diffusion. Then how do they go in? This is the third part. In the third part it reads, if not, they don't go. So if not, then how do they cross the plasma membrane? So our third point is going to be, these polar molecules are transported by carrier proteins. So polar molecules are transported by or with the help of carrier proteins. And carrier proteins means they will move from the peripheral proteins or extrinsic proteins, they will bind to these substances, they will come in energy requirement, or it could be through those protein channels also. So this type of movement is active. So in our answers, in our answer for this question, all three parts should be answered. First part, movement of neutral substances by diffusion, which is a passive process in which the solute moves from higher to lower concentration. Second part, polar molecules cannot cross the membrane in the same process. Third part, how do they move in? They move in with the help of proteins, which are carrier proteins. So all three parts of the question must be answered. Seventh question. Question number 7. Name two organelles that are double membrane bound. Part 1 of this question. What are the characteristics of these two organelles? This is part 2. State their functions and draw labeled diagram of both. Let us answer the first one. We have done all different organelles. What are the organelles? The structures, the membrane bound structures which are present between the plasma membrane and nuclear membrane, that is in the cytoplasmic region. So there are two which are double membraned. So first part, the two double membraned organelle are 1. Mitochondria and 2. Plastids or chloroplasts. So we have named the two organelles which have double membrane. What are the characteristics of these two? So now let us do mitochondria and chloroplast. What is the characteristic? In case of this mitochondria double membrane, outer membrane smooth, inner membrane folded. These folds are known as cristae. In case of chloroplast, both the membranes are smooth. So both membranes are smooth after the chloroplast is completely formed. Next characteristic, in case of mitochondria, there are F0, F1 particles, F0, F1 particles or oxysomes which help in ATP synthesis. In case of chloroplast, there are thylakoids which are arranged in the form of grana. So, grana which help in photosynthesis. So, function wise, mitochondria is known as the powerhouse of the cell and chloroplast is the food factory of the cell. One more difference if you have to talk of, mitochondria are present in both plant and animal cells, whereas chloroplasts are present only in plant cells. Now, let us just quickly draw a small diagram. We have discussed the structure in detail, but when we draw mitochondria, normally we take the oval shape, outer membrane smooth, inner membrane thrown into finger-like folds, which are known as cristae. It has its own circular genetic material. There are ribosomes which are free in the matrix, and on the crista are these F0, F1 particles. Don't forget to label these, the membranes. These bulb-like structures are oxysomes. This is the DNA, and these dots which are visible here, these are ribosomes. And we know in mitochondria and chloroplast both, the ribosomes are 70S type. Let us come to the diagram of chloroplast. Again, we prefer drawing the most common shape, which is oval, double membrane. Inner membrane is also smooth, there is no fold. Folds were there when we said the chloroplast is being formed, but after it is fully formed, we don't see those things. And these sac-like structures, which are called thylakoids, they are arranged in the form of a stack called granum, and there are multiple grana. These grana are interconnected plus connected to the inner membrane. It also has its circular DNA. In the stroma, ribosomes are there. So these again, these are ribosomes. The matrix which is here is known as stroma. This structure is a granum, double membrane. All other labels we have already discussed, like these connecting lamellae are known as stroma lamellae. DNA should be labeled here. So now we have answered all parts of this question. This was two organelles which have double membrane, so we have named these two. Then the characteristic features of the two. They share very many similar characteristics, like both have DNA, both have 70S, and structurally diagrams of both. In case of mitochondria, F1, F0 particles, folds in the membrane, inner membrane, DNA and ribosomes. In case of chloroplast, grana, connecting lamellae, that is stroma lamellae, its own DNA, ribosomes, and the matrix is known as stroma. So all parts discussed. So that means in question number 7, we have discussed all three parts. Now let us take question number 8.